Hello everyone, today we are going to talk about placenta and umbilical cord. These are the two temporal transient structures and they are no longer needed just after the birth, but nowadays they are considered not as biological wastes just after the birth, but as the valuable source of stem cells. So today we are going to discuss about the structure, function during the pregnancy and what we can obtain from these structures just after the birth. So let's start. First, we have to recollect something from the first semester, and this is exactly the moment of implantation. To understand what is the placenta and its structure, first we should remember how the embryo get implanted into the maternal uterus. So here we can see the embryo and the endometrium. As we know from the last lecture, in the endometrium there is the function layer and the basal layer. And exactly the function layer is used by the embryo as the soil. You know, if we put a seed in the soil, it forms roots. In the same way, if embryo invades the endometrium, it forms roots and starts to get some nutrients out of there. So how those roots are formed and the steps of this process we are going to discuss today. So you remember that embryo on this stage of development is represented by the outer cell mass or the trophoblast. It is of green color here, and the embryoblast or the inner cell mass. And embryoblast splits into two, the epiblast and the hypoblast. And the trophoblast is also represented by two layers. The outermost layer is formed by fusion of cells. So this is a multinucleated structure, which is called syncytial trophoblast. Here the name is syncytial. So syn means common, cytio means cells. So the fusion of cells is implied. So here the cells are fused. In the cytotrophoblast, cells are still bordered one from another. Okay, so exactly trophoblast is the subject of our interest during this lecture. So from its name, trophoblast, tropho means nourishment. It provides nutrients to the embryo. How? First of all, in the outer layer, in the syncytial trophoblast, some spaces called lacuna appears. Empty spaces, here they are. And eventually, with the time, they get fused with the maternal vessels in the endometrium. So here we can see the next stage. Those lacuna, they become filled with maternal blood, which is discharged from the maternal vessels in the endometrium. So starting from this stage, embryo gets nutrients from maternal blood. And there is the exchange of gases through the trophoblast. Then, if at the beginning of its formation, chorion is represented just by cytotrophoblast and syncytial trophoblast, later on, it forms root-like structures called chorionic villi, invading deep inside the endometrium. As embryo enlarges, it needs more nutrients, so villi become larger, more prominent, more developed. And there are three generations of villi. First of all, villi, root-like structures, are represented only by cytotrophoblast, surrounded by the syncytial trophoblast. And this is what we can see at the beginning of the placenta formation. Then, inside those villi, some mesenchymal cells appear, mesenchymal cells of fetal, of embryonic region. And such villi are called secondary. They appear relatively later. Then, at last, on the 21st day, we can see mature villi, tertiary villi, with blood vessels inside them. So villi are washed by maternal blood from outside, and inside they contain fetal blood vessels. So, the structure of the mature villi, of the tertiary villi, includes blood vessels, mesenchymal tissue, cytotrophoblast, and syncytiotrophoblast. Okay, then talking about villi, talking about the mesenchymal core, we have to say that among those cells, one population is especially important. These are the Hofbauer cells. They have also the name placental macrophages or fetal placental antigen presenting cells. So Hofbauer cell is widely spread. What you should remember and keep in mind that these are the cells of fetal origin. Of course, they are inside the chorionic villi. Then they are antigen presenting cells, so their function is to engulf the antigen and to present it on their surface. So they have a major histocompatibility type 2 receptors there, and they are more common in early placenta, but what has the clinical aspect is that such cells could be used to transfer different types of viruses. So such viruses as human immunodeficiency virus could be detected in placenta exactly in the Hofbauer cells. So they can destroy some pathogens while they can sequester some other pathogens and helping them to escape from the immunity. Okay, let's move on. So placenta. What is placenta? After we have discussed the chorion, types of chorionic villi, now we have to say that placenta is made up of two parts. The fetal is represented by chorionic villi, and they invade the endometrium of the uterus. So taken together, endometrium of the uterus, together with those chorionic villi, they form placenta. Something like if we take the layer of the soil together with roots of different plants, this will be placenta. Roots like chorionic villi, soil like the functional layer of placenta. Now, during pregnancy, the endometrium is called decidua in Latin. So if you see the word decidua, you imply endometrium. And we have to understand that not whole endometrium participates in the formation of placenta, only locally, where chorion invades it. So there are three regions of endometrium or decidua you have to distinguish. First, look at this image, here they are shown. Endometrium is shown with orange color, and you can see that here endometrium, which is invaded by chorionic villi, and this is what we call the decidua basalis. Decidua basalis. Then the layer of endometrium surrounding the embryo, projecting inside the uterine cavity, is called decidua capsularis, like a capsule around the embryo. It is devoid of chorionic villi. Actually, they are present, but they are not very well developed. Then, and decidua parietalis. This part of endometrium lines the uterine cavity from inside. So those three parts, basalis, parietalis, and capsularis, they all are formed from the endometrium, from its functional layer, but they differ in the function. So, in the formation of placenta, only decidua basalis takes part. Okay, later on, as you can see, embryo enlarges and amnion get fused with the decidua. So they become fused one with another. Okay, let's continue. Then also several terms you have to remember. This is the chorion frondosum of villus chorion and chorion lever. So locally, where in the place where chorion invades the decidua basalis, we can see the so-called villus chorion. Villus chorion, or the chorion frondosum, so-called. This is the highly vascularized part of the chorion. Then chorion lever is the opposite side of the chorion, which is faced to the decidua capsularis. If we return the previous slide, so here should be the villus chorion, and here should be the chorion lever, or bolt chorion, like that. So it's very easy to remember. Of course, if chorion is in contact with the decidua basalis, here close to the maternal vessels, so villi are more prominent, and this part we call villus chorion. Opposite side, where chorion is faced to the decidua capsularis, there is the uterine cavity, no blood vessels. So this part of chorion is bald and it is called chorion leaven. Also, one more term should be keep in mind. This is the chorionic plate. Chorionic plate here. This is the layer of placenta from which the villi project. So here is the chorionic plate. Then, talking about placenta, also another term you should remember and to define. This is the placental barrier. Placental barrier is the barrier between the fetal and maternal blood. They never mix one with another, and they are separated by the barrier, made up of those components you have to remember. Why this barrier is important? Because it helps to protect fetus from some kinds of pathogens. Not all, of course, and lots of medicines, lots of drugs, lots of chemicals, poisons, they cross this barrier. Because this barrier provides not only gases to the fetus, but also some nutrients, rather large molecules. So together with them, lots of different molecules can pass. So, of course, during pregnancy, maternal organs should be protected and different exposure to different pollutions should be avoided. So let's talk about this barrier. What separates the two vessels? First of all, this is the layer of syncytial trophoblast around the villi. This is the first barrier, and it is faced to the maternal blood. Here, maternal blood is around the villi. Then, the layer of cytotrophoblast, it becomes discontinuous till the end of the pregnancy, but still it performs the function of the barrier. Then, basal amino of the trophoblast, here in the diagram, basal amino of the trophoblast is shown in a dark pink color. Then, connective tissue, mesenchymal tissue of the villus, it is shown here, and a very important role in this layer, Hofbauer cells play, those macrophages of fetal origin. So when we are talking about this connective tissue of the villus, we should imply macrophages, providing immune surveillance. Then, basal amino of the endothelium and the endothelium of the blood vessels. This is the typical structure of the blood capillary inside embryonic villi. So here, basal amino and endothelial cells. And all those elements, they separate maternal blood from the fetal. Also here, you should remember what is the syncytial nodes. Here they are shown, another term should be memorized. These are the clusters of nuclei of trophoblast cells, and they become more prominent again at the end of the pregnancy and could be used to estimate the term of the pregnancy. So syncytium, the syncytial trophoblast at the end of the pregnancy forms syncytial nodes. Okay, what also should be mentioned and what you have to distinguish, these are the types of the villi. Some villi, they comes in contact with the decidua, with the site of endometrium. So such villi are called anchoring villi. Some of them are free-floating and they are based in the maternal blood and they are washed by the maternal blood from different sites. Yes, besides what is also should be mentioned is that the trophoblastic shell is formed. Okay, it is not shown, I'm sorry. Okay, another terms, three terms you should keep in mind. Inside placenta, fibrinoid deposits could be found, and those deposits are represented by fibrin and some dead placental cells. And there are three types of fibrinoids. They were discovered in the same institute in the 19th century in Germany, and those three scientists, they were working together and they discovered that one layer of fibrinoid is located just under the chorionic plate, and it is called Langhans fibrinoid. Then another two fibrinoids, fibrinoids of Rohr and Nitterbuch, they are located on the border between the fetal part and maternal part. So maternal part fibrinoid is represented predominantly by maternal decidual tissues, and fetal part of fibrinoid aura is represented by the cytotrophoblast cells and predominantly of fetal origin. Okay, just remember those three fibrinoids and the regions where they could be found. Then, yeah, besides the fibrinoid of Nitabuch is exactly the layer which is the borderline where placenta separates from the endometrium during the parturition. Then another term to be understood, this is the cotyledon. If we look at the fetal part of the placenta, so here is the part from the amniotic surface, and here is the surface of the placenta which is faced to the maternal endometrium. We'll find out that this fetal part of placenta is separated into regions because fetal part it is separated by the septa in the maternal part, and those septa they form some regions in the fetal part. So those regions are called cotyledons. Cotyledons, these structures could be distinguishable with the unarmed eye. Just remember. Yeah, and now we have to say that placenta is hormone-producing organ, and those hormones are very important. First, it produces estrogens, but very important to understand that it doesn't have the full set of enzymes to provide estrogen synthesis, and for this purpose, to produce estrogens, placenta uses the hormones produced by the fetal adrenal cortex, so they should work one with another to produce estrogens. Then, human chorionic gonadotropin. I'm sure you are aware about this hormone. This hormone is important to maintain the yellow body existence at the beginning of pregnancy, and also in maintaining the endometrium during the pregnancy. So this is the hormone which is estimated in the blood, in the urine, and helps to detect pregnancy by pregnancy test. Then human chorionic somatomammatropin or human placental lactogen is called lactogen or mammotropin because it stimulates the mammary duct proliferation, so it prepares mammary gland for the lactation, future lactation. Then relaxin prepares cervix for the delivery. Then leptin, you know, it is also produced in adults, but during the fetal period of life, it regulates the level of nutrients, nutrient supply to the fetus. So, yeah, okay, these are the general hormones. Of course, there are many more produced by placenta, but you should remember at least this. We consider them as one of the most important. Besides, human chorionic gonadotropin is resembling luteinizing hormone and also thyroid stimulating hormone. So it affects the thyroid gland and it should be also keep in mind. Then, what about the placenta, the specimen of placenta? What you should distinguish here? In some specimens of placenta, we can find amnion attached to it. So if we look this amnion with this epithelium, this line of epithelium, then we can understand that this is the fetal part of placenta. This is the part of placenta which is faced to the fetus. Really, in their life, the amnion and the placenta, the chorionic plate, they get fused, and really we should separate them if we want to detach amnion from placenta. But when specimen is prepared, amnion sometimes is detached. So in the real life, they are attached, they are fused. Then what we can find in the placenta? Chorionic plate, then decidual cells. Here they are. Decidual cells, these are the cells of the functional layer of endometrium. And during pregnancy, they become large. As you can see, we call them decidual cells. Then fibrinoid of the Langhans, exactly here, it is present of pink color. Then these are the villi, And what we can find inside villi, we can find fetal vessels. So now we can conclude this is the tertiary villi. Then, except vessels, they are surrounded by the mesenchymal tissue, and also they are surrounded by the trophoblast. Notice that trophoblast forms syncytial nodes we were talking at the beginning. So these are the clusters of nuclei. That's what we can distinguish. Then also we can find maternal blood. And here in this cavity, maternal blood, which is washing those villi from outside. And we can see that exactly here is the barrier between the fetal vessels and maternal blood. Blood-placenta barrier or placental barrier. So these are the structures we can find in the specimen of placenta. Let's move on. Now let's talk about umbilical cord, in brief and the most important. First, the structure of the umbilical cord. The outer surface is covered by the amniotic epithelium. It is continuous with the amnion, of course. Then under this, we can find the mesenchymal connective tissue, which is called Wharton's jelly. Wharton is the name of the author. So Wharton's jelly could be also subdivided into subamniotic, so just under the amniotic epithelium, then intermediate and perivascular, surrounding the vessels which are present inside the umbilical cord. And we have to say, very important, that we consider here umbilical cord lining, which is represented by the superficial layer of the Wharton's jelly and amniotic epithelium. Taken together, they form this umbilical cord lining. Why it is important? Because it is used in stem cell therapy. So lots of stem cells could be obtained out of there, so just remember. And because if we detach the amniotic epithelium, a layer of mesenchymal cells is detached together with the epithelium, so they are considered as a distinct structure. Then, inside umbilical cord, and it's very important, 3 vessels are located. 3. 2 arteries and 1 vein. Usually, you know, everything's quite opposite. 1 artery surrounded by 2 veins. But here, opposite is true. 2 arteries and 1 vein. And everything's even more complicated because arteries, they carry deoxygenated blood from the fetus, while the vein carries oxygenated blood to the fetus. Okay, and that's what we are going to discuss later. What we can find in the umbilical cord except arteries, except vessels. Also, the remnant of allantois is present. You remember allantois, this is the structure which was used to provide vessels to the placenta. But itself, allantois, it represents the rudimental structure, and it is present only in the umbilical cord close to the fetus. Also, yolk sac could be found also there, but it is very short. Not so prominent as allantois, but also could be found there. Then, what else? Here the specimen is given, and we can see macroscopically that here is the umbilical cord lining and vessels present inside the umbilical cord. So, here the 2 arteries, and in the center 1 vein. Here is the example of the MCQ from the USMLE database. And the question is, just after the delivery, the umbilical cord is analyzed, and it was revealed that only 2 vessels are present instead of 3. And 1 vessel contains 90% of oxygenated blood. Then another vessel contains only 50% oxygenated, the blood oxygenated only for 50%. So the question is, which of the following describes the normal function of the missing umbilical cord structure? So out of 3 vessels, 1 is absent. First, you should understand which vessel is absent. Then you have to answer what function it performs here. So, first of all, you should remember that there is 1 vein in the umbilical cord and 2 arteries. So vein carries oxygenated blood, arteries deoxygenated. So from this information we can understand that one vessel carries more oxygenated blood, much more oxygenated than another. So we assume that this is an artery and a vein. I'm sorry, I'm confused here. So we assume that this is the vein with oxygenated blood, and this is an artery, the artery. So another artery should be absent, but actually everything's even more simple. Of course, if there is no vein in the umbilical cord, such delivery could not result in the birth of normal fetus, because vein brings oxygenated blood. So one might conclude that in any way the vein is present in the umbilical cord, so the missing vessel should be on the artery. So everything's very easy if you understand and if you are not confused. There are lots of subjects for confusion. One vein to arteries, and vein brings oxygenated blood, while arteries deoxygenate. Just remember that. Okay, and at the end of this brief lecture, I want to give some information about the fetal stem cells, as I consider them as very important. They are already widely used in practice, and as future doctors and future parents, you should understand what is the role of those cells and what is the perspectives for their use in practice. So, such structures as umbilical cord, here it is, then placenta and amnion are considered as biological wastes, yes, and they are discharged as just after the birth. But at the same time, they can be used to obtain stem cells. And now we are going to talk what kind of stem cells we can obtain and what we can do with them, for what. So first of all, and the most well-known stem cells obtained from the umbilical cord, these are the hematopoietic stem cells, and they are already used. They are approved by governments of many countries, so I think you are aware, and several words we'll discuss later about them. And then also, what are the stem cells are usually not known by the majority of people. So we also should mention that there are some mesenchymal stromal cells. Sometimes they're called mesenchymal stem cells. These are terminological differences, but mesenchymal stromal stem cells could be found in all those three parts, amnion, umbilical cord, and placenta, but this is a very heterogeneous group of cells, and their potential differ depending on the place from which we obtain it. Even if we obtain mesenchymal stem or stromal cells in the umbilical cord close to placenta or far from placenta, close to fetus, their properties would differ. So the interesting population, but heterogeneous. Then epithelial stem cells, they can be obtained from the amnion and umbilical cord lining, the outer surface of the umbilical cord. Actually, this is the same, because umbilical cord is lined with the epithelium, which is continuous with the amnion. Here you can see amnion is continuous with the umbilical cord lining. So these epithelial cells theoretically are very prospective, because if you remember from the beginning of our subject, the amnion derives from the epiblast, and epiblast gives rise to all three germ layers, ectoderm, mesoderm, and the endoderm. So amniotic cells, they are close relatives of the epiblast, and if they make only one step backwards, they can recapitulate all the potential of the epiblast, and theoretically we can obtain derivatives of all three germ layers out of the amniotic epithelium stem cells. And in vitro, such experiments are more or less successful, but the problem is that we have to distinguish stem cells in the amniotic epithelium from differentiated cells, and it is very difficult to do, because stem cells, they are very heterogeneous, and it is very difficult to distinguish them from others, but they are very prospective. And research in this field very promising. Then chorionic stem cells also could be found in the chorion, and decidual stem cells also could be found in the decidua, in the maternal part of placenta, but they are not so prospective as those above mentioned. Why I didn't include hematopoietic stem cells in the most prospective? Because they are already successfully used in the practice, and also we'll talk about them later. But first, let's talk about fetal stem cells advantages. Why these cells are more beneficial than other kind of stem cells. First of all, adult stem cells. Fetal stem cells. Fetal stem cells, there is a lack of ethical aspects, so they can be used without any ethical problems. For example, we don't need to invade the organism to obtain them, because placenta and umbilical cord, they are discharged and they are not needed by the fetus or the newborn child, so there is no ethical aspect in collecting them. For example, if we want to obtain adult stem cells, we need to invade the adult organism. We need to obtain some adipose tissue or hair or other tissues out of the adult organism. So, next, low content of somatic mutations. Obvious, because just at the moment of birth, we have less number of somatic mutations than at the moment of aging. In other terms, a newborn organism contains less mutations than adults. Then there is no risk of teratoma formation. We know that embryonic stem cells, this is the inner cell mass of the embryoblast. If we take embryoblast itself and put it in the live organism, in another organism, embryoblast or embryonic stem cells, they form teratomas, such special type of tumors. But fetal stem cells, they are obtained at the moment of birth, so they are much more differentiated, and they do not form teratoma. So there is no risk of such tumor appearance. And this is the advantage of fetal stem cells if we compare them with the embryonic stem cells. Then low immunogenicity. That's why fetal stem cells are not so readily rejected as other types of stem cells, because in their surface we can see low MHC type 1 receptors content, so the major histocompatibility complex receptors are not very well expressed, and also because they expose HLA-G and E antigens, which are of tolerogenic effect, so they stimulate the formation of immunological tolerance towards them if they are put in the organism of the recipient. That's why umbilical cord blood is transfused more easily than the blood of the adult person, and stem cells obtained from the bone marrow of adults. Okay, so, but some disadvantages also should be mentioned. First of all, the relatively low amount of stem cells in one sample. If we collect the umbilical cord blood, for example, nearly 100 milliliters could be obtained. And despite there is a relatively high content of stem cells comparing with adult blood, but in any way, 100 milliliters is a very small portion if it is transfused to adults. So several umbilical cord units should be used if to treat in case of treatment of adult person. That's why the umbilical cord blood is very expensive. That we'll talk about later. Then financial issues. Also, the umbilical cord blood or any other fetal stem cells should be paid because they should be preserved in special cryobanks in a liquid nitrogen, usually under very low temperatures. They are frozen, and of course they could be sequestered there for a long period of time during the whole life of the patient and even more, but along this period they should be paid. And this is a disadvantage if to compare with adult stem cells, because adult stem cells, they can be obtained directly from the patient along the whole life, and some portion of stem cells could be obtained. But of course, with age, the number and amount of quality of adult stem cells decreases. That's why fetal stem cells are more advantageous in case we are talking about treatment of the aged patient. Okay, so that's what we have to say about the advantages and disadvantages of fetal stem cells. And now, why umbilical cord blood is not so perspective as one might think. Here, the two dynamics are shown. This is the dynamics of cord blood units which are preserved every year. So, how many cord bloods are sequestered in cryobanks? And another diagram showing us how many cord blood samples are requested by patients. So here, how many cord blood samples are sequestered, and here how many cord blood samples are needed. And you can see the difference. 700,000 are sequestered, while less than 5,000 are needed. The difference is huge. So much more cord blood is preserved than it is needed. And during the last years, we can see decline in the demand. Why? Because really last several years, the procedure of obtaining stem cells out of adult bone marrow was facilitated significantly. So nowadays, the cost of adult hematopoietic stem cells is less than the price of the umbilical cord blood stem cells. That's why this type of cells is not very perspective in future, despite it is still widely used. And another diagram I want to show you is that nowadays cord blood stored in the private cryobanks is used not only to transfuse the hematopoietic stem cells, but also to treat different other diseases, for example, neurological disorders. So what is more perspective for the umbilical cord and other fetal stem cells, other sources of stem cells in future? The most perspective is to use not umbilical cord blood, but other parts of fetal tissues like umbilical cord lining, maybe amniotic epithelium. It is better to store, it is better to keep in the cryobank, because in the future, when regenerative medicine would invent special procedures how to use those stem cells to treat different types of disorders, they will be already preserved and ready to use, like that. So this, I think, is the last slide of our lecture. So thank you for the attention. Subscribe our channel, and thank you for watching.